Just so you know, I did pull them off to the side for you to, in case you forget the specific ones. I gave you the enzyme, so a decarboxylase, the vitamin associated with this is going to be thymine, and the coenzyme associated with that is TPP. Okay? So in case you forget, like if they ask about a specific enzyme and you forgot what coenzyme goes with that, you were like, oh, it's a decarboxylase. I know it's going to be thymine. Okay? Just to reiterate it. Okay? So make sure you know those. We're going to re-go over this. It's really important that you know the vitamins and the coenzymes because they love those questions.
And they didn't do a lot of that last time, so hopefully it'll come up this time.
So make sure you know the difference between a coenzyme and a cofactor. Okay? Cofactors are required for the activation of an enzyme. They're going to be more mineral-based, so calcium, potassium, copper, zinc, things like that. Okay? Yes? Everybody recognize the beautiful little chart on the bottom? Anybody know what it's called? That's the name of the chart too. So remember, this is the rate. This tells you the rate of your reaction, right? So this is, the bottom part is your straight concentration. The side is your velocity. So your Km is going to be when the substrate concentration is at the point where you need half of your maximum velocity. Okay, so Km is half Vmax. Yes? Okay. Remember, that means Km is a substrate concentration. A lot of people forget that. Okay. So if my Km goes down, what does that tell me? That my Vmax has gone up. What does that tell me about the relationship between my enzyme and my substrate? The avidity is really high, right? Because it takes less substrate to get to my max velocity. Does that make sense? So that attraction is going to be much greater, thus I have a faster reaction. Okay? Does that make sense to everybody?
Cam goes down.
Cam goes down. So, what is the difference between a competitive, you guys remember these competitive and non-competitive? Yes, okay. So if I'm saying competitive, what stays the same in this reaction? The Vmax is going to stay the same, but my Km is going to what? Increase. What about non-competitive? Yep, my Km stays the same, but my Vmax actually decreases. Okay, make sure you know this. All right. So transport proteins. Okay, the biggest one we like to talk about is hemoglobin. So myoglobin transports what? Oxygen. How many oxygen? 1. How about hemoglobin? How many oxygen? 4, right? And it is what type of protein hemoglobin is? Quaternary, yeah, because it's got 4 subunits, remember? 2 alphas, 2 betas. Does everybody remember that? Yes. Okay, if you don't, please review it. There's a little bit of information here about it. So hemoglobin transports oxygen. Sometimes it transports carbon dioxide. Okay, structures 4 protein chains, 2 alpha, 2 beta. Each unit has a heme group. Each heme group has an iron, okay? And each iron will bind 1 oxygen. Does everybody understand that? Yes? What other thing did we talk about is made up of heme groups? Huh? No? Well, we're talking about hemoglobin. Oh. How about cytochromes? Cytochromes made of heme groups? And we're talking about electrotransport chains, cytochromes made of heme groups. Yes, it's all on the notes, I promise you. Okay, cytochromes are also made of heme groups. Oxyhemoglobin is going to be oxygenated hemoglobin, okay? It means it is carrying oxygen from the lungs to the periphery, okay? Carboxyhemoglobin is when it's combined with carbon dioxide, and it is transporting back from the periphery back to the lungs. The way that you would normally think about it, we breathe in air, we get oxygen from it, it gets into our blood, and it goes out to everything, okay? The blood comes back, less oxygen, back to our lungs where we pick up more oxygen. Does that make sense? I mean, that's pretty standard. Okay. So once you bind 1 oxygen to hemoglobin, what happens after that? it's easier to bind. So the second, third, and fourth ones bind a lot quicker, okay? I gave you the different forms of iron just so you know this is a reiteration of stuff we've already covered, but just so you know the ferrous iron, which is the form that's absorbed and carried with the hemoglobin, ferritin is your storage form of iron, and transferrin is your transport form of iron. Okay? Everybody recognize the chart at the top? A little bit? This is the Bohr effect. I'm going to make this chart super easy to understand. Okay? So, if we're starting at this point right here, think of this point as your fingers and your toes. So this is your periphery. We go over here, we're at your lungs. Okay? So, on this end, we have a low partial pressure of oxygen. Okay? So we're going to release oxygen. Does that make sense? It's kind of like we don't have a lot of oxygen or blood in our fingers and toes, we're going to let it go. As we go this way in our chart, okay, our partial pressure of oxygen goes up, right? We're going to bind oxygen. Does that make sense? So there's not a lot of oxygen in your blood, your body releases it. That's going to be your finger and your toes. There's a lot of oxygen, which is going to be in your lungs, your body is going to bind it and carry it back out. Does that make sense? Yes? The different sides, so if we're going up, we have an increased affinity of binding when we have a higher pH. So the up area is for a high pH. The decreased affinity is for a low pH. Yes? Good? All right, so we've done this with carbohydrates and with lipids so far. We're going to talk about protein metabolism. So we're going to eat food, which some of you are doing right now. Okay? Food gets in the stomach. We have movement through the stomach. What happens in the stomach? Does anybody know? Okay. So we release gastrin, right? What does gastrin do? Gastrin is going to stimulate your chief cells and your parietal cells. Yes? Yeah? Okay. What do your chief cells do? They release pepsinogen. Yeah? So everybody understand that? And then what do your parietal cells do? HCl. Okay? So this is going to help digest food, right? Okay. Your pepsinogen needs that HCl because the HCl helps convert the pepsinogen to pepsin. Does everybody understand that? Once pepsin is made, this is autocatalytic, which means once pepsinogen, it converts pepsin, now that pepsin will keep converting. So you'll get more and more pepsin. Does that make sense to everybody? Okay. You laugh? Okay. Does anybody know what type of enzyme pepsinogen is? It's got a very specific name. It's called a what? Where's we'll ask you this? It starts with a Z. It's a zymogen. Does everybody remember hearing that word before? Zymogen means that it is a precursor to an active enzyme. Okay? So I gave you a whole list of them. All of these are zymogens. Okay? So trypsinogen, proelastase, chymotrypsinogen, so keep going. These are your active forms. Okay? Yes? So you have all these enzymes that are floating around. If there's no food, they're not going to do anything. So once there's food in there, we start this conversion, they get converted to an active form, then they start breaking down food. Does that make sense? Now we're not eating away our body all the time. Got to do that. So just like we talked about when we talked about carbohydrates and lipids, CCK is going to help the conversion of all of these. Okay? Yes? And release. Everybody good on that? Very similar to what we talked about before. We talked about carbohydrates and lipids. Really similar. I gave you all of these so you know what they are and what they cleave. Okay? Because remember, we're breaking down protein, so we're going to cleave amino acids off. Does everybody understand that? Yes? Okay. So I gave you all of the very specific ones. So, urea cycle. See, I only had 1 page for protein. So that was really easy, right? Because we had like 2 pages for all the others. Proteins are the easiest ones. So let's talk about urea. So we have all of these amino acids floating around, and what do we have attached to all of our amino acids? One of our side groups is a what? Nitrogen. And nitrogen is not good when there's a lot of it in your system. So what do we got to do with it? We're gonna get rid of it, right? So most of the nitrogen in your system floats around as glutamate, okay? We all know glutamate is what? It's an amino acid, but what else about it? It's an excitatory neurotransmitter. Make sure you remember that. So glutamate is gonna be inside the mitochondria, and it's gonna be converted to alpha ketoglutarate, and we're gonna get a what off of that? We're gonna pull the nitrogens off of that, okay? So then we're gonna convert to carbamyl phosphate, okay? This requires ATP, this first step, okay? Of all your enzymes over here, by the way. So that first enzyme is CPS1. That requires 2 ATP. So getting rid of the nitrogen does require energy, okay? So once that carbamyl phosphate is made, we're going to convert it to citrulline. And just so you know, if you look at the list of your alternative amino acids, this is on that list. Okay, so this is kind of like a alternative amino acid. Okay. So the enzyme that does that is ornithine transcarbamylase, which you can OTC, which is a lot easier. But they will write enzymes all the way out, just so you guys know, just to confuse you. Okay. Once it's converted that, we can't do anything else in the mitochondria. So we're going to, because this amino acid is pretty small, we can just get right out of the mitochondria. Okay. Now this is where we start getting interesting. We're going to take citrulline and we're going to convert it to arginosuccinate. We're going to use another amino acid for that. Does everybody see that? Okay. So now we have used how many amino acids?
3. Because we have the alternative.
Okay. How many regular amino acids? 2, right? So we've used glutamate and aspartate. Make sure you know that. We're using amino acids and we're going to make some too. Okay? So arginosuccinate, okay? This is your first A1. This is arginosuccinate synthetase. Really easy. We're making arginosuccinate. Easy one to remember. We're going to convert arginosuccinate to arginine. That's our amino acid we're making. Okay? Argine's amino acid. Okay? This requires fumarate. This is going to be arginosuccinate ligase. So basically, we're cutting off that succinate section of it, leaving arginine. Okay? From arginine, we're going to convert back to ornithine, and we're going to get urea off of that. Okay? What I want you to grasp is this is, you got to know this whole thing. Make sure you know the amino acids that are associated with this. Okay? The ones that are made, the ones that are used. Okay? Make sure you know your enzymes. Specifically know which ones are in the mitochondria and which ones are not in the mitochondria. Okay? It's really pretty easy though, because all the ones that are not in the mitochondria, start with A. I'll start with A. Yes.
Can we take pictures of this or?
It's actually on Schoology. The whole entire packet is on Schoology. Yep. So I uploaded it a while ago, and I think most of y'all have the code, right? So you can get the code from someone, they'll get it, and you can print it off if you want to. Okay. Let's talk about enzymes a little bit farther. So we're going to talk about transaminase enzymes, okay? Transaminase enzymes means we're going to take a substrate and we're going to convert it to a what? Amino acid or back and forth, right? Okay? I'll give you a bunch of examples here. Just make sure these are found in the liver and they require B6. Okay? We're going to talk about specific ones. So AST and ALT, have y'all heard those? In another class, right? We talk about liver function, correct? Yes? Okay, so let's talk about the box on the side. So transaminase reactions, these are your reversible reactions. So alanine plus an alpha keto acid gives you pyruvate and glutamate, okay? This is ALT. Okay? Alanine, ALT. It's alanine transaminase is what that stands for. Okay? So if we're going to go to aspartate plus a keto acid, gives me oxaloacetate and glutamate. What do you think that one is? AST. It's pretty easy, right? So you notice, though, that we're making things that can feed into other places, right? So what does that tell us? If we eat a lot of protein, we can use that protein once it's broken down into amino acids to make energy. Do you see that? Because pyruvate can feed into what? Where can pyruvate feed into? If you're going to make energy. TCA cycle. What about oxaloacetate? TCA cycle. Does that make sense to everybody? So we can make energy from protein. Okay? These are deamination reactions, okay? This is gonna be direct release of ammonia. So glutamate plus NAD gives me an alpha keto acid plus ammonia plus NADH. See, we're actually making energy there too. Glutamine plus water equals glutamate plus ammonia. Asparagine plus water equals aspartate plus ammonia. Good? Yes? And just to make that conversion a little bit easier, this chart shows you where all of your amino acids can flow into your other cycles to make energy. That easy. So look at all of the amino acids that can be used to make transaminated into pyruvate. How about into acetyl-CoA? You can see you can use all protein that you eat can be used to make energy. Good? You don't have to eat carbs for energy. Okay. Make sure you know the difference between ones that are ketogenic and ones that are glucogenic. Okay? These ones that I've listed right here, you can see glucogenic and ketogenic are all these top ones. Just ketogenic ones are these bottom ones. Okay? Yes? The chart can be overwhelming if you haven't studied your amino acids yet. All right, let's start with the fun stuff. All right, let's talk about vitamins. Okay, so, and you can't see that. So vitamins. We're going to start B1. Vitamin B1 is thymine. Make sure you know the source of this. Okay, important. You need to know the sources. So we're going to talk about pork and organ meat. The coenzyme for thymine is what? TPP, what stands for what? Thymine pyrophosphate, okay? This functions reactions involving the removal of carbon dioxide. Okay, just to reiterate, this is a decarboxylation reaction. It was in that little chart on the other page, but I've tried to give it to you a couple times. Okay? So you make sure you know you cannot metabolize carbohydrates without vitamin B1. Without TPP. Does everybody understand that? Because what reaction requires this? Does anybody remember? Big reaction. Requires TPP. Pyruvate to acetyl-CoA. So if you can only do glycolysis, you're not really getting that much ATP, right? So if you can't go through the rest of the cycle to metabolize your carbohydrates, you're not going to get a lot of energy. Everybody remember that? Pyruvate dehydrogenase is that huge reaction, has a lot of coenzymes. Yeah? It's on the chart with the TCA. Okay. Deficiencies, very, okay? There are 3 types, dry, wet, and infantile. Make sure you know those. And Wernicke-Korsakoff's, and everybody knows that this is mostly associated with alcoholics, right? Good. Okay. Make sure you know the deficiencies, too, that are associated with individual vitamins. B2 would be riboflavin, so make sure you know the source of these. And these coenzymes are going to be flavin, so your FMN, your FADs, okay? Talked a lot about these when we talked about the ETC cycle, when we talked about TCA cycle. Yeah, you heard all these words. So these are involved in dehydrogenase enzymes. Yes? Okay, so I just put on there oil ring, just so you know. I don't know if y'all learned it that way, but so just see the loss or gain of electrons. So deficiencies in vitamin B2 are going to be red and lit, so the corners cracked. And then sometimes you'll have yellow vitamin, you'll have yellow urine. Sometimes you get a deficiency in this. A lot of that's associated with dehydration. Yeah. It's usually associated with dehydration. It can be excess aid. Yeah. It's actually. So the main one that they ask about is the red and lit cracked at the corners. Vitamin B3, niacin. So NAD, NADP, NADP. What pathway? Pentose-phosphate pathways. Sure. All right, so oxidative reduction reactions, dehydrogenase enzymes. Deficiencies are going to be pellagra. Does everybody remember what this is? Yes? Deficiencies, B3, niacin. So these are your technically 4 Ds. Give you 3. So dermatitis, diarrhea, and dementia. The 4th one would be death. It's a big deal. You don't want to have too little B3. It's important. How many reactions do we talk about had NAD in it? A lot. Okay. Toxicity can cause red flushing. If you've ever taken niacin, you know it feels like your whole entire head is on fire. I've only had this reaction by accident once. I did not take it. It was on something that I ingested. So, and it will feel like a flush. Usually people who work out a lot will take this. Yeah?
The natural antibiotic.
Yeah. Like it's like really like your head's on fire. So B5 is going to be pantothenic acid. We're going to think source again. So meats, fish, poultry. Notice most of these sources are protein-based. Yes. Okay. Coenzyme is going to be a contingent of coenzyme A, which we all know when we think of acetyl-CoA, right? When you hear coenzyme A, you should think acetyl-CoA and fat, right? Okay. So this is a thioester bond, part of your acyl groups. A lot of that is going to be your fat. Okay. Deficiencies, fatigue, sleep disturbances, impaired coordination, nausea. Okay. B6, pyridoxine, so meat, poultry. This has veggies and seeds. Coenzyme is PPP. Okay. This one is involved in amino acid and glycogen metabolism. Okay. It is required, I just said this a minute ago, for transaminase reactions. Remember, I said without B6, no transaminase reactions? Okay. Okay. Deficiencies, irritability, convulsions, muscular twitching. You can have a deficiency in this if you take an oral contraceptive. Okay. It can actually affect your B6 absorption. Deficiency. Okay. B9, folic acid. So your coenzyme is going to be THF. Okay. Your AKA for it is FH4. Okay. Vitamin C and NADP are required for the formation of the THF. That's important to know. It's got the extra star by it. So functions, it aids in the formation of purines and pyrimidines. So RNA and DNA, right? That's why it's really important if you're pregnant and building a baby that your RNA and DNA is being made in sequence correctly. Otherwise your baby might not function. So involved in the maturation of red blood cells too. So that's really important. So both of those, you can see how it relates to pregnancy right there. Deficiencies in this can cause megaloblastic anemia. So you can have large immature red blood cells. It is very similar to the look of B12 anemia, but it's not exactly the same. So it can mimic it. So make sure you know the difference between the two for boards. All right, so tropical sprue, pregnancy in women, and GI issues, red tongue. You have a really red tongue if you have a deficiency. Tropical sprue. Huh? No, that's not tropical.
Is it just like megaloblastic anemia?
No, it's not. It's similar, but it's usually more in tropical areas. Yeah.
The question they had associated with this one, on the last one, was which vitamin is associated with spinal bifida.
But that would make sense, because we talked about DNA and RNA and red blood cells and all that stuff. So you think building a new body in your body.
But, yeah.
So you should think formation of a human. You need this for it, right? Yeah. Biotin. So this is the only one that we don't start right off, except for B9 with meat, but legumes, veggies. This is going to be carboxylase reactions. It's required for fat synthesis, amino acid metabolism, glycogen formation. Yeah. You can have a deficiency if you eat a lot of raw eggs. Please tell me you do not eat a lot of raw eggs, but it can interfere with biotin absorption. Okay. If you have a deficiency, fatigue, depression, nausea, really generalized symptoms. Okay. Muscle pain. All right. B12. Okay. You all know this one. So muscle meat, fish, eggs. Make sure that you know that B12 helps with the transfer of a single carbon unit. That's why it's really important. Okay? So think about formation, maturation of red blood cells, helps with peripheral ring for hemoglobin. It's needed for methyl group transfers, which is that single carbon unit. Okay? Deficiencies, lack of intrinsic factor. Who has this? Who lacks intrinsic factor? Huh?
Pernicious anemia, or people that are vegetarian.
People that are vegetarians, alcoholics. How about people that take a protein pump inhibitor? People who have chronic heartburn and take those protein pump inhibitors, do they have low intrinsic factor? Yeah, because they don't have what? What do you need for intrinsic factor? Huh? You need intrinsic factor for B12. How about HCl? You need acid in your stomach. If you're taking something all the time that tells your body to stop producing acid, you're not going to have enough intrinsic factor to absorb B12. Yes? Okay. Megaloblastic anemia, if you have, and this is that pernicious anemia, if you have pernicious anemia because you don't have enough B12, it will turn into megaloblastic anemia. Yes? Vitamin C, so this one should be really easy for everybody. So we talked about hydroxylation reactions. Remember we talked about collagen and elastin, okay? Maintains cartilage, bone, dentin, important for collagen synthesis, helps make steroids in the adrenal cortex, helps absorb iron in the GI tract, helps convert folic acid to FH4. We just talked about it a second ago in the last slide. Okay? It's an antioxidant for cytoplasm. Make sure you know the difference. Remember we talked about vitamin E, which is the antioxidant for what?
The nucleus.
So, yeah. We talked about one that was inside, one that was outside. And it's on the slide when we talk about the lipid-soluble vitamins. It's on there. So deficiencies, please. I know this seems outdated. Scurvy, so degeneration of teeth, skin, blood vessels. And that is because if we can't make collagen, what happens? That stuff breaks down, right? Degeneration of teeth, skin, blood vessels. You think collagen for all of those things, right? Gums. So you can think pyrite for that if you want to. You can have a toxicity of vitamin C, and it can cause kidney stones. Okay? Good? Make sure you're getting enough vitamin C. Important. All right, let's talk about, so we're going to move from vitamins to minerals. Okay? So, iodine. Okay? We should associate this with thyroid hormones, right? Yes? We have a deficiency in iodine. What can we have? A goiter, right? We have toxicity of iodine. What can it do to your thyroid? It can suppress the activity for toxicity. A deficiency causes a goiter. Okay? Copper. So we think about enzymes that are associated with iron metabolism when you think of copper. Okay? A deficiency, so if we're associating the enzymes that are associated with iron metabolism with copper that's needed. If we have a deficiency in copper, it can cause an anemia. Does that make sense? Yeah. Okay. Can also cause changes. If you have a toxicity, what is this called? Wilson's disease. Everybody knows. Right?
Oh, another one of the toxicity for, no, deficiency for iodine that they asked was creatism, but only children.
Yeah. I don't go over every single thing. I think that's an overlap between another class too.
Yeah, and I think that's why it was on there last time.
Chromium, so think things like enzymes that are associated with glucose and energy metabolism, okay? So if you have a deficiency, obviously you're going to have impaired ability to metabolize glucose. If you have a toxicity, it can actually inhibit enzymes, cause kidney and skin damage. And this is usually an occupational exposure. Like maybe someone who works in a factory that puts chrome on stuff, that kind of thing. Someone who works on cars a lot. Okay? Yes? All right, calcium. Please think bone, tooth formation. We should also think blood clotting, right? Think about those calcium bridges when we talked about vitamin K, yes? And the cascade, okay? So deficiency, stunted growth, rickets, osteoporosis, convulsions. Those should be pretty easy. Phosphorus, we're going to think, again, bone, tooth formation. Deficiencies, a big one you need to know about is demineralization. So if you have a deficiency in phosphorus, you can have a demineralization, okay? If you have a toxicity of phosphorus, it can cause something called phossy jaw, which is erosion of the jaw, okay? Potassium, this is going to be fluid balance. We think sodium, potassium pump. We think Remember, if you excrete sodium, you're going to keep potassium. If you keep sodium, you're going to excrete potassium. We talked about your corticosteroids and stuff like that, right? Okay? So fluid balance deficiencies are going to be muscle cramps. If you have a deficiency in potassium, if it's really bad, it can be life-threatening, okay? Because it's going to affect your heart at that point, okay? Toxicity, if your kidney's not functioning, it can cause cardiac arrhythmias. The reason I said kidney is because we talk about kidney, when we talk about potassium, we talk about keeping sodium, getting rid of potassium, right? Aldosterone, y'all remember that stuff? Whenever? Okay. Sulfur, you get most of your sulfur from food preservatives. I hate to say that, but this is associated with acid-base balance. It's very unlikely that you're going to have a deficiency of sulfur in your system. But if you did, what amino acid would you associate with it? Cysteine, because we couldn't make a disulfide bond if we didn't have enough sulfur, right? Yeah? Chlorine, so this is important for extracellular fluids. If you have anything associated with it, you're going to have too much chlorine, and it's going to be associated with high blood pressure, so think salt. Sodium chloride, okay? Magnesium, enzymes that are involved with protein synthesis, okay? Deficiency is going to be growth failure or behavioral disturbances, okay? Muscle cramping is also one that's associated with this. So if you have deficiency in magnesium, you can have a lot of muscle cramping too. Toxicity is diarrhea. Zinc, so I put down every time like what enzymes it's involved in, okay? So enzymes involved in digestion, so deficiency would be growth failure. You can have small sex glands if you have a deficiency in zinc. Toxicity would be fever, nausea, vomiting, or diarrhea. Okay? Sodium, so we all know this is acid-base balance, fluid balance, nerve function, okay? If we have low sodium, we have muscle cramps, mental apathy, reduced appetite. If you have too much sodium, what does it cause? High blood pressure, because we're keeping fluid, right? Where the sodium goes, the fluid goes. So trace irons, okay, functions for hemoglobin, energy metabolism, because of a heme associated with cytochromes in the electron transport chain, right? So a deficiency in iron would cause what? Anemia. Toxicity in iron would cause siderosis, and it can contribute to cirrhosis of the liver. Okay? Selenium is associated with what vitamin? E. And does selenium need vitamin E to function as an antioxidant? Yes. Does vitamin E need selenium to function as an antioxidant? No. Good. So deficiency in selenium can cause an anemia, and why is that? Because vitamin E protects what? Red blood cells. Cell membranes, all that stuff. Yes? Y'all remember that? It's on the slide, vitamin E. Okay? So if you have too much selenium, GI issues, lung irritation, fluoride, probably what we have in our water that we're drinking. So most of your source of drinking water, tea, seafood, supports bone structure. If you have a deficiency, it can cause tooth decay. If you have too much, it can cause molting of the teeth or increased bone density. I think everybody's heard my voice and touch like that. Yes?
Yes.
That is all of them, guys. All the slides. It's a lot of slides. Okay, so I want everybody to pull out their phones. Yeah, I'm going to ask you to pull your phones out. Seriously. I'll get a pen if you know. So get your phone ready. This is the pen. You're going to go to Kahoot.com and it'll ask you to put a pen in. Yep. Tell me when y'all are ready. So right now I have no players. Kahoot.it might work too. Yeah, if you go to Google and type in Kahoot, it normally comes up right away. We won't make it to all of them. As we go through these, I want you to make a nice little chart for yourself on a piece of paper or somewhere. and write C, make a line, write L, make a line, and write P, make a line. If you get one wrong, I want you to determine if it's a carbohydrate question, a lipid question, or a protein question, and just tally them. So you know how many get wrong in each section, so that way you know which ones you need to look at a little bit more. So I split everything up by carbohydrates, by lipids, by proteins, just to make it easy. We got 3 players.
Drip assignment.
You normally only have, I mean, it'll ask, does it ask you to give a nickname or something
Yeah.
like that? Yeah, it asks for like a nickname or something. I don't really care what name you use. I mean, you could just give yourself a letter. You don't have to play, just to give you a good idea of how many questions this is a huge mix of questions, okay? This covers everything. Remember, my review is just a review, okay? So there was a lot of information that you should already know that I just kind of skimmed. Everybody ready to go? I think it'll let you add as you go, too. Okay. So right now, there's 6 panelists. Which protein is associated with Alzheimer's? You get 20 seconds. I guess we're supposed to. It's going to give you the answers. Amyloid. We're going to go to the next one, okay? We're going to do these stats. This would be a protein question. Okay? That's a protein question too. Just let you know what they are as we go.
I have a question on that question. Why wouldn't it be hydrogen bonds?
Yeah.
Because hydrogen bonds are associated with all protein structures, not just one. So that's going to be the structure between the tertiary, the alpha and beta, attaching to each other, not just hydrogen bonds. It's kind of a psychology question. It does come up with a biochem question. Plasmid reticulum. Most of y'all know that. These are kind of how Board's going to work questions. Promise we've gone over all of these. Or you have them as a review. This is on that lovely big chart that I gave y'all. Not all of them are going to be hard. And excuse my spelling mistake. I hope we went over this one. It's supposed to be to the tissue. Remember, increase 2,3-BPG increases oxygen binding. We went over that one. It's on the page when we went over glycolysis. I went over that 2,3-BPG goes 2,3-BPG increases oxygen binding. So if it increases oxygen binding, it's going to facilitate the travel of oxygen to your tissues. Yes. I'm trying to word things so that it's close to kind of how Board words it, or a little bit better. Remember, we just went over biotin. Remember, we talked about fats. If you looked at the functions, that's on there.
I put oxidation, instead of
hemoblast anemia and a hemolytic anemia. Make sure you know the difference. I know these seem really difficult now. This is why I want y'all, and we're gonna do more of these, okay, because I have a bunch of them. But just kind of get used to how questions are asked too. We just went over this, so I don't expect you to absolutely know the answer. And we just went over this, it's okay. Like it's one of those tiny little squares. So four of y'all got it. We're gonna have a full entire meet together. We're gonna go just through questions. It does help you start to think. See, I can write that on mine. It does help you start to think about though how the questions are worded and what they're associated with. First thing we went over this. We went specifically over disaccharides, monosaccharides. Remember, amylase is a very long, lots and lots of monosaccharides. All the others are just two monosaccharides together. This is all in the notes. Infomers. And I put that in the notes for you, worded exactly like that. We just went over Simon's. I know we just went over this. Yes. So we can get pepsin, we get procarboxylase from that, we can get chymotrypsin. The lactase is the only one that's on Simon's.
When we do this next time, can we do it without music?
Yes. I thought we clicked no music when we first started, but it didn't listen.
It was cool for like 2.
Sorry.
And then I was like, nope.
Absolutely, we can do no music. So this is a crossover question between another class, another part of boards.
Actually, no idea.
I have no idea what that was.
I've never, yeah.
Heard of that?
I have it in my notes, yeah. Yeah. I like crossover questions better, because the words like crossover questions. It is in your notes. Which is arachidonic acid. It's in there. Some of this we actually went over today. I mean, not just that, it's some of it. Remember 2, so it's going to have 2 alpha, 2 beta. That's going to be 4 subunits. Quaternary structure. So only when we talk about this, a quaternary structure. Probably is mathematical question. At least I hope. Please don't overthink it. That should be 4. Sorry, it should be 4. But it should be 4. I don't know why you did that. Yeah, no, it should be 4.
Don't overthink it.
I don't know why I did that, but I promise it is 4. Any nerve tissue, think peripheral neuropathy, think issues with your eyes, yeah. We'll do a couple more, and then we'll just start where we left off next time. Just kind of really look over your notes for next time.
Whenever you want to schedule that.
We're going to talk about it in a second. We'll make this our last one. This is also in your notes. It's in your notes. The initial 3-cursor anchor protein, it's 3 units of glucose together. It's called glycogenin. It's in your notes. That's where we start. Then UDP glucose is how we add from that point on. Okay. I think that was 20. Okay. We're going to start there next time. Okay? Did y'all like that?
Yeah.
Or did it make you cry?
Yeah.
Okay, we're both. We're both.
Are those just random questions?
There's 22 questions. Girls.
It's a combination of lots of stuff. I have probably a bank of like 3-4 questions. Just from lots of different things, different sources. Some of them, I just sit and flip through your notes and look at the questions. I'm like, oh, that would be a good question to ask. That would be a good question to ask. Okay, when are we going to meet next? Everybody look at your stuff. So you guys have boards. When?
2 weeks. 19 days. 18 and